The topic I shall be dealing today about is infection. So the first thing that comes to your mind about infection is the microorganism. Exactly. It is a relationship of the host or the animal body with the infecting microorganisms. So all the microorganisms are not pathogenic in nature. Some of the organisms can be beneficial to the human, whereas some organisms are pathogenic in nature. So in medical microbiology, we'll be dealing all about the organisms that are involved in the pathogenesis or disease causation. So infection is interaction between the human host and the infecting microorganism or pathogenic microorganism that has the ability to cause an infection in a human body. Now, depending upon the relationship of the organism with the host, we can classify the parasite or the pathogenic organism into two types. One is called as a saprophyte, the other one is called as a parasite. Now, coming to the distinguishing features of these parasites or the saprophytes is that saprophytes are free living and they usually depend upon the dead and decaying organic matter. Because they do not have the ability to multiply in these conditions, they do not cause infections in the body. Whereas parasites are the one that are pathogenic in nature and disease causing agents. So saprophytes are the one that are free living, can be seen in the soil and the water and mostly seen on the dead or decaying organic matter, and they are the one that are involved in the degradation of the organic matter. Think that if these saprophytes are not present in the environment, then there would be no decaying of the dead or decayed organism. So in that way, these saprophytes are helpful to the environment by decaying the organic matter which are present in the environment and completely rely upon the dead and decayed organic matter. And these organisms are the one that cannot cause infection because they do not have the ability to multiply in this dead and decaying organic matter. And moreover, there is an exception where you can see that organism which is called as Bacillus subtilis. It is called as opportunistic pathogen. So this organism usually cause infection in the person who is immunocompromised, where the immunity or the immune power in an individual comes down. In that case, this organism is able to cause an infection. So such type of organisms are called as opportunistic pathogens. So saprophytes sometimes can act as opportunistic pathogen, otherwise they do not cause infection in a dead and decaying organic matter and the human body. Now coming to the parasites, as I told you, they are pathogenic in nature. They have the ability to establish and multiply in a host. So they can cause infection either by multiplication in a particular area of the human body or by producing certain types of products which are released inside the human body and cause the infection. Example I can give about this is the toxins. So these organisms which are parasites can establish themselves in the human body in two ways. One is called as a pathogen, the other one is called as a commensal. So among these two, it is the pathogen that is usually the one that is involved in the disease causation or which causes a pathogenic disease in the human body. So they produce a disease wherever they get established and multiply themselves. Whereas commensals are the one that lives in harmony with the human host. That is, they do not cause infection, but they give certain types of beneficial advantages to the human body when they are present in the human body, but do not cause infection. But there comes a time when these commensals can also cause infection, and hence they are called as facultative pathogen. And it is only in the case where the immunity of a person usually comes down or in an immunocompromised patients. So very good example of that is you can say the patient suffering from Mycobacterium tuberculosis. So in this patient, the immunity completely comes down. So in this case, the normal flora or the commensals which we call, they usually cause an infection. Otherwise, they are in commensalism with the human body. They live in harmony with the human body. They do not cause infection to the human body. So parasites can be in a relationship with the host in two ways. One is called as a pathogen, the other one is called as a commensal. And commensal sometimes can be a facultative pathogen. Otherwise, the pathogens are the one that causes the infection. So the main the problem in medical microbiology is to deal with the pathogenic organism to find a way to treat this kind of organism that produces diseases in the human body. Now coming to the classification of the infection. So before going on with the infection, let me give you a clear detail about what is an infection and what is an infectious disease. So infection is the lodgement and multiplication of the organism microorganism or the pathogenic microorganism on the human host, and it need not lead to an infectious disease until and unless the organism is in more amount, more dosage, and it is producing certain types of toxic products that usually harms the patient. So in that case, the infection can lead to an infectious disease, but it need not be that all infections are infectious diseases. So the basic difference between an infection and infectious disease is infection will not lead to infectious disease until and unless certain criteria is fulfilled by the microorganism. Now coming to the classification of the infections, you can classify infection based upon variety of characteristics. First I will deal with the primary infection. So primary infection is the immediate infection that we get when we interact with the pathogenic microorganism is called as a primary infection. That is, initial exposure to a microorganism is called as a primary infection. Now coming to the reinfection, it is the subsequent infection with the same type of organism on the later time is called as a reinfection. That is, the same pathogenic organism which has caused a primary infection, if it infects the human body, is called as a reinfection. Next is the secondary infection. Now there is a difference between reinfection and a secondary infection. Secondary infection is the one that is usually defines that when a person is suffering from a particular disease, now if he gets infected with another type of disease, that means he is already suffering from a disease and infected by other type of disease also, then it is called as a secondary infection. This is can be very well explained again with the help of the Mycobacterium tuberculosis infection, where the immunity comes down and it leads to the secondary infection by the other type of organisms. Now coming to the focal infection, also called as a focal sepsis, where the organism or the pathogenic organism multiplies at a particular area and then cause the generalized effect. Generalized means the whole body gets affected after the initial multiplication at a particular area. Take for example tonsils. So initially it is the only one area which gets infected with the pathogenic organism, and if it is not treated properly, then it can lead to generalized infection where the whole body can be infected when the organism travels along the blood and causes different types of infection of the different types of bodies. So that is called as a focal infection. That is, starts with the focal at one particular point and then becomes generalized if not treated properly. Next, coming to the cross infection, again similar to that of the secondary infection, where the person is already suffering from a disease and he gets a new disease from the external source. So a good example of this is the nosocomial infections. Nosocomial infection is nothing but a person who is being treated for a particular disease in a hospital gets another type of disease from this hospital. That is a bacterial infection. The person is suffering from a particular infection and he gets a bacterial infection from the hospital, then it is called as cross infection. So there is a difference between secondary infection and a cross infection. So here we have a new set of infection from the external source. Nosocomial infections are the one that a person or a patient gets when he is in the hospital. So hospital induced infection can also be called as nosocomial infections. Now next, coming to iatrogenic infections. So they are called as physician induced infections. So it is the physician that induces the infection to the patient while treatment or giving certain types of drugs or while catheterization or dialysis. So these type of infections that are induced by the physician during the treatment process are called as iatrogenic infections. Next, coming to the source of infection on the human body, it can be classified as either exogenous or endogenous. So from the external source it is called as the exogenous infection. From the internal source they are called as endogenous infections. Now coming to the exposure of the symptoms, that is how a person exposes his symptoms. Depending on that, you can say that the symptoms can be apparent or inapparent or subclinical infection. That is, the person is suffering from a disease but he is not expressing the symptoms. In that case he is called as it is called as an inapparent or subclinical infection. So here the person can also act as a carrier to carry the diseases to different person while he is in contact with the other person. So now coming to the atypical infection. So here in this case the typical characteristics of the infections are not seen in a particular patient. So take for example if a person is suffering from a particular bacterial disease. So the bacterial disease has certain types of symptoms. So that cannot be expressed, that typical characteristics cannot be expressed in that case case of atypical infection. Now next coming to the type of classification is a latent infection. In this kind of infection, the person who's already suffered from an infection, has overcome from that infection, harbors the pathogen in his body for a longer period of time. So later in part of his life, these symptoms or this disease can also come up and cause the infection in the person. So the person who harbors the infective organism for a longer period of time in his body is called as a latent infection. So this can be like the person exposed to a particular type of infection at the initial time, like suffered from that, and he has overcome that infection and still has a infecting organism in his body, and later on in his period of life he can express that type of infection again. So that is called as a latent infection, mostly seen in HIV patients. So initially they get attacked with the HIV virus, they show certain symptoms, but this virus becomes latent, and on exposure to the later part of the life, this infective organism can develop the infection in a human body. Now next coming to the sources of human infection. Now what are the different sources from which a human can get the diseases from the external sources? So first source of infection is the human itself. So human can like carry the organism with himself and can transmit the diseases from one person to the other person. So the first source of infection are the humans themselves. So they can be a patient or they can be carrier. So patients are the one who are suffering from the infection, and carriers are the one that carries the infections but do not show the symptoms that are related to the infection. So based upon the exposure of the symptoms, you can classify the carriers into various types. So carriers can be classified as healthy carriers and convalescent carriers. So healthy carriers are the one that carries the infection but they are not getting that particular infection, whereas the convalescent carriers are the one that has already suffered from that particular disease and they carry the organism and they can transmit the organism to the other humans. Now depending upon the duration of carriage, how long do they carry this organism with them, so you can classify them as temporary carriers or the chronic carriers. Temporary carriers are the one that can carry the organism for a period of about only 6 months, whereas if they carrying it for a longer period of time, more than years together or for the rest of their life, they are called as chronic carriers. So they are the one that can cause the infection to any type of the human when they come in contact, and depending upon how they transmit the infection, they are called as contact carriers and paradoxal carriers. So contact carriers are the one that can directly transmit the infection to the human in which they are coming in contact, and paradoxical carriers are the one that usually gets the infection from the carriers. So this is how the humans themselves act as a source of infection to transmit the disease from one human to the other human. Now coming to the other source of infection are the animals. So the animals can also act as a source of infection, and the infection that we get from the animals are called as zoonotic infections or zoonosis. So here the domestic animals or the rodents are the one that can usually cause spread the infection. So if they are not showing any type of the symptoms that are present related to their particular organism, they are called as asymptomatic or they are called as reservoir host of that particular organism. So when they come in contact with the human, they are able to spread this infection from the animals to the humans. So the diseases that are transmitted by the animals are called as zoonotic diseases, and the examples can be of different type where different organisms are involved to transmit the infection. So in case of bacterial infections are usually transmitted from the rats to the disease is a plague that can be transmitted to the human, and it is a viral disease that is a rabies that is transmitted by the dog bite. When the dog bites and injects the organism subcutaneously into the human body, this can lead to the rabies diseases in the human. Next is the protozoal disease that is transmitted by the cats and the organism is toxoplasmosis. The disease is toxoplasmosis. And coming to the elements, they are transmitted by the dogs and the disease is hydatid. And finally the fungal disease, that is zoophilic dermatophyte, which is transmitted by the cats and dogs. So these animals can act as a a reservoir host. They did not show the symptoms. They have the pathogen with them, and they can transmit this to the human when they come in contact with the human. So the diseases caused or transmitted by the animals are called as zoonotic diseases. So this is a secondary source of infections that transmit the diseases to the human body. Next, coming to the third type of source of infection is the insects. So insects, mostly the blood sucking insects are the one that can transmit the disease from one person to the other person, and the insect borne disease are called as arthropod infection. So it is, if it is an animal, then it is called as zoonotic infection, and if it is through the insect, it is called as arthropod borne infections. And here also the insect acts as a vectors. They act as a reservoir host for different types of pathogen, and they can transmit it to the human body. So based upon the different types of insects involved, you can classify them as mechanical vectors and biological vectors. So in the mechanical vectors, example, the disease of dysentery or the typhoid, which can be transmitted by the domestic flies. So that is, they only act to transmit the infection from the foot to the humans. Whereas in the biological vectors, these organism enters the insect, and they are a part of the developmental cycle of the insect, and they are usually transmitted from the insect to the human blood. So example is the malaria you can take. So the protozoa usually multiplies in the it acts as a developmental, it is one of the developmental cycle that undergoes in the mosquito, and these sporozoites are usually like transmitted to the human body, where they usually show the symptoms related to the malaria. So one of the developmental cycle is usually present in the mosquito. So this usually transmits the infection to the human. So they are called as biological vectors, where the pathogen acts as a present in the insect acts is one of the developmental cycle of the insect, and they are transmitted from the insect to the human body. And there is incubation period, germination incubation period, that is usually seen in the insect, where the entry of the organism in the insect until they form the developmental cycle, complete development take place in the insect, it is called as the incubation period. So after the developmental cycle, they are matured, pathogenic organisms are introduced into the human body to cause the infection, and this most of the insects acts as a reservoir host for most of the type of infections. Now next source of infection is the soil and the water. So it is an external environment which is contaminated with different types of microorganisms. Now coming to the soil, this soil may harbor different types of pathogenic bacteria, usually the sporulating bacteria that can gain an entry into the human when the human usually consumes the food which is contaminated with the soil particles and all. So the best example of the infections that we get from the soil is from the Clostridium tetani, that is a tetanus. So the sporulating organism survives in the soil, and they can transmit the infection to the human. Apart from that, the fungal infection can also be transmitted. One is example is Histoplasma capsulatum and Nocardia asteroides. And apart from that, you can also see the parasites such as roundworm and hookworm that are usually seen in the soil, and when this contaminates the food or the vegetation, they can be transmitted to the human. So they are a very good source of infections. Next, coming to the water. Water, again, depending upon the type of the organism that is present, you can say that it is either contaminated with the organism, or it is the aquatic vectors that contains the microorganisms that can transmit the infection. So contaminating vectors, you have this cholera bacteria, and you have the other protozoals that are usually present in the water that contaminates the water, and when this is consumed by the human, then can lead to infection. Now, aquatic vectors are the one that carries the insects that carries the microorganism, and that can be transmitted to the human. So it is the carriers, you can say. Aquatic vectors are the carriers of infection, whereas the contaminating vectors are the one that are contaminates that water, and when consumed can cause the infection. Next coming to the food. So food can be contaminated with a microorganism, or they can be a pre-existing infection in the food with the different types of microorganisms. So we have a staphylococcal infection that is coming due to the food poisoning, that is due to the external contamination. Here the organism usually produces certain toxins and releases it into the food, and when we consume that food, we land up to the food poisoning. Now we have the pre-existing infection is from the salmonella organism that usually have the ability to multiply in the meat or certain food products. So when we consume this kind of food, then we can end up to different types of infection. So food can also act as a good source of infection in human body. Now this is about the sources of infection, that is how we get the infection from the different source. Now let us see how these infection can be transmitted from one person to the other person and one source to the other source. So here on this we can classify different types. So first is we can deal about contact. So contact can be a direct contact or an indirect contact. So in the direct contact, the best example that can be given is about the sexually transmitted disease, where the example is the syphilis or the gonorrhea that are usually transmitted during sexual contact. So that is a direct contact where the organism can be transmitted from one patient or one person to the other person. Now coming to the indirect contact, it is through the fomites or the toys. Examples can be the toys or the pencils that children use. So there's an infection called as diphtheria that can be transmitted in the school children when they are sharing the pencils. So based upon that, you can classify the contact into direct contact and indirect contact. Now coming to the inhalation, most of the respiratory diseases are usually caused during the inhalation process. So organisms are usually present in the environment, and when gains an entry via the respiratory pathway, they can land up to different types of diseases. So inhalation can cause like different types of respiratory diseases, mostly related with the diphtheria or mycobacterium, tuberculosis, etc. So when inhaled, they can cause the different types of infection. So ingestion lands up to intestinal diseases, where this transmission can take place either via the food or the water or the hand-borne diseases. So food-borne diseases such as we have salmonellosis, or we have streptococcal diseases or the staphylococcal toxin diseases, and then we have waterborne diseases where cholera and etc. can be transmitted via the water that is contaminated, and hand-borne diseases usually the dysentery. And then next type of transmission is inoculation. Inoculation is direct establishment of the organism into the particular tissue of the human body. Usually takes place in case of wounds. So when a wound is caused in the human body, this can lead to landing up of the different types of organisms. So bacteria that can land up and multiply in the wound diseases are mostly the anaerobic forms of organism, that is Clostridium tetani. And we have the viruses that take for example the rabies virus can be directly placed while the dog is biting in a subcutaneous form. So the organism can be directly placed in the wound, such as the virus of rabies. And then we have the other type of transmission via the insects, where blood sucking insects already dealt with that they are able to carry the organism as a biological vectors and can transmit it to the human when they bite or when they suck the blood from the human body. So during that course they can inoculate the organism into the human body, and the organism can be seen in the blood of the human body. Next is congenital or tetragenic, that is called as a vertical transmission, that is from the mother to the fetus. So the organism has the ability to get transmitted from the mother to the fetus and cause different types of infection in the fetus, can lead to the stillborn or death of the fetus also. If the child is surviving from that particular infection, then there might be many disformities in the child that is being born. So mostly during the first trimester, when the person is the pregnant lady is infected with the rubella, in the first trimester there is a possibility that this organism will interfere with the organogenesis of the child, and hence there is a deformity in the child that is present in the fetus or the child that is usually born. So that is called as a vertical transmission, because the transmission takes place from the mother to the fetus, and the organism having the ability to cross the placenta. Next is the iatrogenic and the laboratory animals. So iatrogenic are nothing but the physician induced the infections are called as iatrogenic. So during catheterization, during lumbar puncture, they might inoculate the organism into the person if the materials that are used are not sterilized properly. And apart from that, you can see the transmission of HIV and hepatitis B virus also. When there is a blood transfusion, if the blood is not sterile, then it can lead to the diseases such as HIV or the hepatitis virus. So these are the different methods of transmission of infection from one person to the other person. Now let us see what are the factors that usually helps a microorganism to become more pathogenic in nature and cause infections in the human body. So in that regards, I would like to tell about two concepts. One is called as a pathogenicity and the other one is called as a virulence. Pathogenicity is the ability of the microbial species to cause an infection in the human body is called as pathogenicity, whereas virulence is the ability of a strain to cause an infection. So you can also say that virulence is the degree of pathogenicity of a particular organism, and an organism can either be a virulent organism or it can be a avirulent organism. Virulent is the one that usually causes the diseases in the human body. Avirulent are the one that do not cause any type of infections in the human body. So pathogenicity and virulence are the similar terms, but they are defined differently. So pathogenicity is the ability of a microbial species to cause the infection, whereas virulent is a characteristic feature of a strain of that particular species, whether it can be a virulent or an avirulent. And the virulence of a particular strain is not constant, it keeps on changing. As I told you, it can be a virulent or avirulent form, and depending upon the condition in which the organism is entering in that particular environment, it can either be a virulent form or a avirulent form. And the virulence of a particular organism can be enhanced, and the process of enhancement of a virulence is called as exaltation. Exaltation can be done by continuously culturing the organism in a susceptible culture or the host. So in case of viruses, if you want to enhance the virulence of a particular organism, you can continuously transfer or culture it in a susceptible host. And the opposite of exaltation is attenuation, where there is a decrease in the virulence of that particular organism. So organism becomes attenuated when they are continuously transferred or subcultured on a media that is not suitable for that particular environment. And you can change the physical environment also to make the organism attenuated one, and it is the attenuated strains that are mostly used in immunization purpose. So depending upon the characteristic feature of that particular organism, you can either exalt the virulence of an organism, or you can attenuate the organism by removing the pathogenic property of the organism but maintaining the immunogenicity of that organism. So that is a concept that is used in vaccination. So vaccines are nothing but they are antigenic preparation, where the antigenic character that has the ability to cause infection has been removed, but it has the ability to enhance the immune system of a particular patient. So that is the concept that is used in vaccination. So attenuated organisms are mostly used in vaccination preparations. So now the virulence of a particular organism is the sum of the factors of different determinants. So how can you know that a particular organism is virulent or not? It depends on certain characteristic feature of this virulent species. So first determinant is the adhesion. So adhesion is nothing but to attach to the host surface receptor. Only after the attachment is able to cause the infection in any type of human host. So the first determinant of a virulent is the adhesion. So adhesion is the first characteristic feature that has to be satisfied by the pathogenic microorganism to show its pathogenic property. So the adhesions are nothing but there are certain filaments or certain components that are present on the surface of the organism that helps to attach the organism to the human host host or the surface receptors that are present on the human host. And remember that this is a specific reaction. It is not that any type of organism can go and interact with any type of the receptor cell and can cause the infection. So the best example I can give over here is the plant viruses cannot cause infection in the human host because they do not have receptors to go and bind and cause the infection. So it is a specific reaction between a receptor that is present on the human host and a ligand that is present on the microorganism. And these appendages that are useful for attaching to the human host are called as adhesins. And there is a tissue tropism and host specificity that determines to which host they have to go and bind and to which host they can go and cause the infection. And there are different virulence factors that helps the organism to go and bind and initiate the infection in the human host. Virulence factors are nothing but the appendages. All organisms have different types of appendages that helps to go and bind to the human host. Take for example, in the viruses we have a protein coat that goes and binds to the human host, and in the bacteria we have fimbriae or like pili that goes and bind to the human host and can initiate the infection. And these are mostly protein and antigenic in nature. That is, they are made up of proteins, and antigenic means they are able to induce an immune or enhance the immune system of the human body. So based upon this, you can also prepare antibodies and can give as vaccination against that particular organism. So the first determinant is the adhesion. Now next is invasiveness. Invasiveness is the ability of the organism to spread inside the host. That is, once it gets attached to the host, it can multiply and will also have the ability to move to other parts of the body of the host. That is, ability to spread in the host tissue is called as the invasiveness. So here you can classify the organism based upon how invasive it is, how how good it can spread within a particular host. So depending on that, you can classify it as high invasive pathogen. They usually produce generalized lesions, and weak invasive pathogen that produce only localized lesions. And there are organisms that lack invasiveness but also have the ability to be fatal and cause the death of the person. Take for example, the Clostridium tetani causes tetanus, and here the tetanus is caused by toxic products called as toxins that are produced by the organism that can move to different parts of the body and can cause a damage to the human body. So invasiveness is the ability to spread inside the human body. Once it attaches and multiplies in a particular area, it has the ability to move. This is what is the invasiveness. Next is toxinogenicity. So all organisms do not produce disease only after multiplying. So the diseases that are produced by the organism after multiplying in a particular host is called as infectious disease, and the diseases that are caused by the products that are produced by the organism are called as intoxication. So that is infectious diseases, intoxication is completely different. Infectious disease is multiplication of the organism inside the host, and intoxication is nothing but the toxins that are produced by the organism that are usually fatal in nature and pathogenic in nature. So toward this particular toxins, our human body responds by producing antibodies which are called as antitoxins. So toxinogenicity, different organism has ability to produce different types of toxins. So based upon that, you can classify it as exotoxins or endotoxins. So endotoxins means it is a component of the organism itself. When the organism dies, it releases this component, and this has the pathogenic property to cause infection in the human body. So they are called as endotoxins. Whereas exotoxins are metabolic products of the organism that are produced during the metabolic activity, and they have the characteristic feature of causing the causing the infection in the human body. So the difference between exotoxin and endotoxin is that exotoxin is heat labile. It is very sensitive to heat, whereas endotoxin is heat stable, that is a bit resistant to the heat. And it is exotoxin is highly potent, that is even 1 mg of the exotoxin can kill millions of guinea pigs in the experimental animals. So it is highly potent, whereas endotoxin is not very potent, but it can induce fever and can cause a bit type of destructive damage to the human body. Next is the antibodies that are produced against exotoxins are called as toxoids, and here in case of endotoxin we don't have any such toxoids that are usually produced, but it is a component of the organism, as I told you, that is usually infective in nature, and the component is mostly seen in gram-negative organism where lipopolysaccharide layer, an external layer to the cell wall, acts as endotoxic component. And the exotoxin has when they are showing the metabolic activity, when they are usually causing the infection, they releases certain types of pharmacological products that brings about the pharmacological activity. That is, they causes the pathogenesis in the human body by inhibiting certain important functions in the human body. Such type of pharmacological activity is absent in case of endotoxin, and the exotoxin shows typical effects, whereas there is no typical effect that can be seen in the endotoxin. Only the thing that can be seen in the endotoxic infection is the raise of the temperature, that is the fever. That is mostly pyrogenic in nature, and exotoxins are produced mostly by the gram-positive organism, and endotoxin is only characteristic feature of gram-negative organism. The organism which contains LPS or lipopolysaccharide layer has the ability to show the endotoxic activity, whereas gram-positive organisms are many that produces these toxins that brings about the different types of infective diseases in the human body. Example of the exotoxin is diphtheria, bacteria that produces a toxin called as diphtheria that can bring about a damaging effect in the respiratory system of the human body. Now we have other type of the determinant that is specifically present in some kind of organism, which are called as plasmids. Plasmids are extra chromosomal material that are present in the bacteria, and these plasmid usually contain certain genes that are showing highly virulent characteristic features. So the gene expresses certain types of virulent appendages that helps to the organism to colonize in a particular area, multiply and produce a deleterious effect in that particular area. And plasmid are also the one that usually makes the organism resistant towards different types of drugs. So the emergence of multiple drug resistant bacteria is because of the property that is conferred by the plasmid, which has the property to resist different types of drugs, and hence it is becoming difficult for us to treat different types of infections with the drugs. So the plasmid is one of the important virulent factors seen in only few of the gram-negative bacteria. That is, organism which has a plasmid are highly virulent in nature when compared to the organism which do not have the plasmids. Next, coming to the bacteriophages. Bacteriophages are nothing but the viruses which attacks the bacteria. Best example I will give over here is the diphtheria bacteria. Diphtheria bacteria as such is not like pathogenic in nature until and unless it is lysogenized with the bacteriophage. Lysogenized means the virus has two types of cycle when it enters the bacteria. One is called as a lytic cycle, the other one is called as a lysogenic cycle. In case of lytic cycle, the organism takes over the host, that is a bacteria, and it completely kills the bacteria, brings about the lysis of the bacteria. Whereas in lysogenic, the DNA or the RNA of the virus usually gets incorporated in the bacterial chromosome. It lives in harmony with the bacterial chromosome, and it is called as lysogenized bacteria. Now it is this viral nucleic acid that contains the tox plus gene. Now the bacteria which is lysogenized with this bacteriophage having the toxplex gene will have the ability to produce the toxins, and this can cause the infection in the human body. So this is how the bacteriophage can also act as a virulent factor to enhance the virulence in a particular organism. Now next important concept that we will be discussing here is the communicability. That is the ability of the microorganism to survive and spread in a particular population. So this factor, that is a communicability, is not important for the multiplication of organism, but it helps to spread the organism in a particular area, and because of this communicability the organism is able to cause epidemics and pandemics in different areas or even worldwide. So communicable diseases are mostly the respiratory and intestinal diseases, and it is the it is through the communicability that the organism is able to maintain its population for generations and generation together, because it is able to spread in more amount or more number of population. So it is a characteristic feature of the organism to spread in a particular environment, and not a feature of the organism to multiply, or it is an intrinsic property of the organism. It is only to spread and maintain its population in the environment and to survive and distribute itself in the particular environment, and this is the reason that they cause endemic and pandemic diseases. Now coming to the bacterial products, apart from this we have seen the other virulent factor that helps to enhance the pathogenicity of an organism. So apart from that we have certain bacterial products that can prevent the bacteria from getting destroyed from the human immune system. So the organism prevents itself from the death and survives in the human body by producing different types of bacterial products. So one such example is a coagulase produced by staphylococci, which is a thrombin-like substance, and it produces a fibrin clot around itself, and this fibrin clot protects the organism inside itself and prevents the damage of the organism by forming from the immune cells. Next is the fibrinolysin. Fibrinolysin are the protein that degrades the fibrin clot that is formed as a part of human immune system to protect or prevent the spread of the infection. So fibrinolysin breaks the fibrin clot and helps to transmit the infection from one part of the body to the other part. Next is the hyaluronidase. There is a hyaluronic acid that acts as a cement between the two cells. Now the hyaluronidase that is produced by the bacteria cleaves this cement and helps in the spread of the infection from one cell to the other cell, mostly seen in the Clostridium tetani. So when a soldier gets hit with a bullet, what happens is they usually amputate the whole leg if he is suffering from this tetanus infection. So what they do is the organism has the ability to move from one part of the body to the other part by breaking the hyaluronic acid that is present. So the best way is to amputate from the other part, so to prevent the spread of the infection. Now apart from that, the other bacterial products that are produced are leucocidins and hemolysins. Leucocidins are the one that can break down the polymorpholeucocytes and thereby inhibiting the immune cells to act upon the organism and decreases the immune status of the organism or the human host, and hemolysins are nothing but that breaks down the RBCs of the human host. So these are the bacterial products that do not give the characteristic feature of exaltation, but they are the one that prevents the organism from the cells of the immune system. Apart from that, we also have the bacterial appendages that prevents the organism from escaping the phagocytosis. Example is of capsules. Capsulated organisms are said to be more virulent than non-capsulated bacteria because they can prevent phagocytosis by the macrophages. Other type of appendages that are seen in Staphylococcus and Salmonella typhi and E. coli is the VI antigen, K antigen also prevents the phagocytosis from the macrophages, and they also inhibit themselves from killing by complement systems. Next is the infecting dose. The infecting dose is very important for the organism to be pathogenic in nature. It is not one or two organisms that can cause the infection in the human body, but a group of organisms when entered into the body that can initiate the infection. That is called as infective dose. So you can estimate this in terms of minimum infecting dose, MID, or minimum lethal dose. So minimum infecting dose are the organism, a group of organism or a dosage of organism that can initiate the infection. Lethal dose is they are able to cause a severe infections in the body. So nowadays we have standardized the condition by taking up ID in infective dose and lethal dose 50. So they are able to produce the infection in 50% of the human or 50% of the experimental animal when all the conditions are standardized. Now next is the route of infection. There are certain organisms when they gain an entry from a particular area, they start producing the infection. So different organism has different areas where they can go and establish themselves and cause the infection. So it is the route of infection that usually determines whether the organism can survive in that particular area or not. Example, cholera bacteria can be effective only when taken orally. when they are consumed orally, they can show their clinical effects, whereas when introduced subcutaneously, they do not show their clinical effects. So it is the route of infection that also matters a lot to the pathogenesis of the bacteria. Now coming to the type of infectious diseases. So you can classify the infectious diseases like based upon whether it is a localized disease or a generalized disease. So localized disease can be only on the superficial disease, or it can be deep-seated disease. Generalized means it has the ability to involve the whole human system, where the organism once multiplies at a particular area can gain an entry into the blood, and the presence of bacteria in the blood is called as bacteremia, and when this bacteria settles in any part of the body can initiate another type of infection. So that is usually producing a generalized effect on the human body. So you can classify as local or a focal infection, that is one area involving one area, or it can be a generalized infection where the whole body is involved to produce the infection. Next we have bacteremia and septicemia. Bacteremia is nothing but the presence of bacteria in the blood. It can be transient, or it can that means the organism can be present only for a few period of time, or in case of chronic diseases you can see the bacteria for a longer period of time. And septicemia is nothing but the organism, once it has multiplied in a particular part, enters into the blood and can initiate infection at the other area. So involving the whole body is septicemia. Next is pyremia, is nothing but the fever inducing property of an organism. So this fever inducing property of an organism again changes depending upon the strain of organism and the species of organism that is involved. And the organism producing different types of diseases, they can be classified as endemic disease, affecting a particular area and particular number of organisms. Best example is a typhoid fever. If a person gets only one area of that particular environment can be infected with that particular disease only for a few period of time. And then we have epidemic disease that is rapid in nature and can spread through a large number of population. Example is influenza. So it is only during the winter season that you can see that the epidemic of influenza can take place. And pandemic is related to epidemic, which is short and involving more rapid, more fatal, and involving whole country, or it can be throughout the world. So that is a pandemic disease. The examples of the pandemic diseases are influenza, plague, cholera, etc. And then we have also prosodromic diseases, that is only related to the cerebrospinal fever such as encephalitis. So only few persons are infected with this particular type of infection. So these are the different types of infectious disease. So in this particular topic we have studied what is infection, and this helps us to categorize organisms and helps in the treatment process.